Welcome back to our series on driving the power of single-cell technologies forward into clinical applications with ScaiVision. I'm Sarah Carl, Director of Data Science at Skylight. If you missed the first video, you might want to go back to it for a quick introduction into single-cell techniques and applications in drug development, as well as an overview of the pre-processing steps required for single-cell data analysis. Today, I'm going to dive into the details of ScaiVision's representation learning algorithm, how it's different from conventional methods, and why it allows for a more targeted approach to answering clinically relevant questions. Let's start with, what is the question that we want to ask of these single-cell data in the context of biomarker discovery? I'd argue that it is, what are the features in this dataset, which cells and which genes within those cells, that allow me to most efficiently separate two or more different classes of patients? This is exactly what representation learning is designed to do. With standard approaches, we are trying to get at this question in a roundabout way. You can't just start taking pairs of individual cells and comparing them to see how they are different, because it's impossible to compare the exact same cell from two individuals. So typically you start by trying to define comparable groups of cells, which could be clusters or annotated cell types, and then you ask what genes are differentially expressed across conditions within each group. But this is really a question about characterizing the biology of those cells in detail, not about separating patient groups. After all, finding statistically significant changes in gene expression does not guarantee that you can use those differences to make predictions. So in the end, you'll get a list of a bunch of differentially expressed genes from different clusters or cell types, but no clear way to use those to define a biomarker or predictive signature. On the other hand, with a supervised learning approach, we take single-cell data with labels, which can be a clinical endpoint, as the input. And models are trained to predict the labels, therefore learning a new representation of the data, which best allows us to separate cells by their labels and make accurate predictions on the sample level. Once we have trained models, we can easily interrogate them to learn which are the features on both a cell and a gene level that were most important for those models to make their predictions. And because the models are designed from the start to learn the best possible separation of samples by label, they're actually very good at ignoring other sources of variability in the data, which could come from either biological or technical effects. To go in a bit more detail into how the model training works, we start with a collection of single-cell samples, each of which can be represented as a matrix of expression values, and each of which is associated with a known endpoint label, such as healthy versus disease. Then we apply subsampling to each sample as a form of data augmentation, effectively increasing the number of samples that can be used to train the network. This allows us to use relatively few biological samples, as the network has the opportunity to learn from many snapshots consisting of only a part of each sample each time. The filters are the part of the network consisting of learnable weights, which form a connection from the gene expression values to the outcome prediction. As the model sees more data, these weights are gradually updated, allowing the network to automatically determine which features are most important for the output prediction. The pooling step allows us to go from cell-level values to sample-level values for each filter. We use a flexible method of summarizing the filter response scores across cells called top K pooling, which takes the mean values over a predefined top percentage of cells. This K value is a hyperparameter and can be varied across different networks that we train. This type of pooling allows for the sensitive detection of disease-relevant cell populations within the sample. After pooling, we have one value for each filter. To create a final output value, the filter values need to be combined in the output layer, which applies a simple matrix multiplication. At this stage, a single output probability is assigned to each possible label class. Altogether, we train the network for a number of epochs, usually around 50. For each batch of input samples, a loss function is used to compare the predicted probabilities with the actual label. We then apply backpropagation through the network and update the weights, with the goal to minimize the loss. This process continues until the loss doesn't decrease any further, and then we consider the network trained. To illustrate the value of this approach in real life, I want to use the example of biomarker discovery in endometriosis. We generated a large single-cell dataset from women both with and without endometriosis, with the goal to discover a diagnostic signature. We'll come back to this project in a later video, so I don't want to go in too much detail here, but I want to emphasize that this dataset had a lot of different sources of variability, which you see summarized in the heat map of PCs on the bottom of the slide. By far the strongest effect driving molecular changes was the menstrual cycle, which was a true biological effect, but not the one we were interested in. You can see this effect in the cycle phase scores on the UMaps here. On top of this effect, we also had a strong sequencing batch effect, which remained to some extent even after data integration. So there was a signal in the data for endometriosis, but it was a weak one compared to all of these other signals. However, with ScaiVision, we still managed to cut through all of this noise and find a predictive signature, which, as you can see here, is actually expressed across multiple cell types. In the next video, I'll continue on the topic of how ScaiVision enables unbiased discovery by retaining single-cell resolution throughout the analysis, with a focus on how we can identify the most predictive cells for a given endpoint without needing clusters or cell type annotations upfront. Hope to see you there.